Greetings, RB Yuki learners. So, like here is a part where we are from doing the detailed structure of the cells. In that, we did mention to say the cell it is a building block of any living organism. So, henceforth, the cell can never be seen using our naked eyes. Hence, it can be seen using the microscope. So in an exam, you can be asked to define what this microscope is. So a microscope, it is any instrument that is used to view smaller objects. It is any instrument that is being used to see or to view smaller objects. That is the function of the microscope. So as I mentioned, every cell can never be seen minus using the microscope. So for us to see the cell, we need to use the microscope. So every cell, it could be a plant cell or an animal cell, can never be seen using or maybe by the means of our naked eyes. They can only be seen using the microscope. So before now we talk about the structure that we can see, I need you to understand that there are three types of microscope that we have. There is number one, there is the handless microscope. Handless microscope. Handless microscope. Now, what is this handless microscope? It's the microscope that we just hold by seeing it. It can be holded using your hand. Okay, so then now we have also what we call the light compound microscope. So the structure that you can see on the board, it is called the light compound what? Microscope. Then lastly, we have what we call the electronic microscope. So electronic microscope also it is the type of the microscope. So now this, whatever you can see on the board, is the one that is being examinable and also that is in your curriculum. So now, according to this, we have the light compound microscope. This is the structure that we can see over the light compound microscope. So over this structure, we have some of the parts which are based with their own function. So we have the eyepiece, we have the barrel, we have the nose, objective lens, stage, the diaphragm, we have also the mirror, we have arm, we have fine adjustment, and also the coarse adjustment knot. So here is a part where we need to understand these part which qualifies that this is the light compound microscope. So let's begin first of all by breaking it down what the eye does. So the eyepiece, what is the function? The function, it is to enable that all the objects are being seen. Okay, so the function it is used to see the smaller objects, or it's used to see the object. The eyepiece, it is used to see the object. Then now we have the barrel, commonly known as the body tube. So the function is to maintain the proper distance between the lenses. It is to maintain the proper distance that is between the what? The lenses. Are we getting the point? So that is the function of what we call the barrel or the body tube of the microscope. Then now we also have the objective lens. So this objective lens, we are saying it is used to magnify the object. It is used to magnify the object. It is used to magnify the object. That is the function of the objective lens. So sometimes in an exam, you might be given some of the words or some of the things that you are required to calculate the magnification part. In that, you must leave that using the x. So x must be represented as you multiply every digit given under the microscope. So that is it. This is an example just showing you. In my next video, I will be doing the calculations on how you calculate differently using the objective lens. Then now we have also the mirror, where we are saying it is used to reflect or to bounce light. So now what is the meaning of reflecting? To reflect simply means to bounce back of light. So like once you insert the mirror, an opaque mirror, the one that is not transparency, the one that is opaque, it will strike light like this, then again it will bend, it will bounce back. the light happens to bounce back from there. So the function of the mirror, it is to reflect light. It is to reflect what? Light. That is the function of this mirror that we can see here. Then now we have what we call the cos adjacent. So all these two, the fine adjacent and the cos adjacent, these are called the nodes. They are nodes. So the fine and the cos adjacent, they are differently shaped. So we can see that the fine adjacent is smaller than the cos adjacent. So what is the function of the cos adjacent? We are saying that it is to move the body tube up and down. It is to move the body tube up and down from there. Then now we have the fine adjacent. The function, they are saying it is used in a focusing of the image with a high power objective lens. It is used in focusing of the image with a high power objective lens. So this one has the low power objective lens, while this has the high power of the objective lens from there. Then now we also have the arm. So the arm, the function, they're saying it is used to support the body tube. So this arm, it is used to support the body tube of the microscope. Lastly, we have what we call the base. We are saying it is used to support the entire microscope. It is used to support the entire microscope. So now here is basically where I need again you to understand some of the functions which I did not mention, which is the nose and also which is the diaphragm. So if you talk about the nose, the function of the nose, you are going to write the function of the nose. It is used to hold the objective lens. It's used to hold the objective what? Lens. It's used to hold the objective lens. That is the function of the node. Now, let us talk about the diaphragm. We're saying it is used to control the amount of light going through the object. That is the function of the diaphragm. It's used to control the amount of light going through the objective going through the object. It is used to control the amount of light going through the what? The object. That is the function of what we call the diaphragm. Okay, so that is it. These are the questions which mostly are affected in biology. Please follow for more, like, and also don't forget to share, subscribe to my YouTube channel, as next I'll be doing the other topics and the other things pertaining to the microscope on how you calculate and invest in over the objective lens. This has been your tutor, Mr. Abi. Bye-bye.